Exact reaction coordinates for flap opening in HIV-1 protease. Shanshan Wu, Huiyu Li, https://orsid.org/000000034079 and Alma https://orsid.org/000000029872 7333 omi@uic.edu. Authors, info and affiliations. Edited by George Schatz, Northwestern University, Evanston, Illinois. Received August 31, 2022. Accepted October 25, 2022. December 2, 2022. Significance. The primary goal of protein studies is to understand how proteins function, which requires understanding the functional dynamics responsible for transitions between different protein conformations. The key to success is to identify the reaction coordinates, the small number of essential coordinates that control functional dynamics, starting with the pioneering paper of Du et al. And seminal works of Chandler Group in late 1990s. Intensive efforts have been devoted to identifying reaction coordinates in complex molecules. This remains a major challenge. Using the recently developed generalized work functional, we successfully identified the reaction coordinates for flap opening of the HIV-protease, an essential process of this complex protein and major drug target. This success marks an important step toward understanding protein functional dynamics. Abstract. The primary goal of protein science is to understand how proteins function, which requires understanding the functional dynamics responsible for transitions between different functional structures of a protein. A central concept is the exact reaction coordinates that can determine the value of committer for any protein configuration, which provide the optimal description of functional dynamics. Despite intensive efforts, identifying the exact reaction coordinates, RCs, in complex molecules remains a formidable challenge. Using the recently developed generalized work functional, we report the discovery of the exact RCs for an important functional process, the flap opening of HIV-1 protease. Our results show that this process has 6 RCs. Each one is a linear combination of 240 backbone dihedrals, providing the precise definition of collectivity and cooperativity in the functional dynamics of a protein. Applying bias potentials along each RC can accelerate flap opening by 2 folds. The success in identifying the RCs of a protein with 198 residues represents a significant progress beyond that of the alanine dipeptide. Currently, the only other complex molecule for which the exact RCs for its conformational changes are known. Our results suggest that the generalized work functional, GWF, might be the fundamental operator of mechanics that controls protein dynamics. Sign up for PNAS alerts. Get alerts for new articles, or get an alert when an article is cited. Proteins are the building blocks of biological systems responsible for most biological functions. Understanding the mechanism of protein function is the primary goal of protein science. The central dogma is that structure determines function. The Holy Grail is the physical principle that governs how the structure of a protein determines its functions. A critical step toward this principle is to recognize that most protein functions, such as ligand binding, allostery, and enzymatic catalysis, involve significant conformational dynamics because a protein has multiple functional structures. For example, inactive versus active conformations, and transitions between these structures are required for its functions. 1, 2. Structure determines function because the specific structure enables the desired functional dynamics. Understanding protein function requires understanding functional dynamics. Importance of reaction coordinates, RCs. Most functional dynamics of proteins are activated processes similar to chemical reactions. A protein must cross an activation barrier much higher than thermal energy to move from the reactant state, for example, the inactive conformation, to the product state, for example, the active conformation. A central concept in activated processes is the RCs, the small number of essential coordinates that fully determine the progress of a reaction. 3. A reaction progresses in the forward direction when RCs move toward the product state and regresses when RCs move toward the reactant state. Movements of all the other coordinates are irrelevant. Answers to the 2 central questions on activated processes, 1. Reaction rate, and 2. Reaction mechanism, hinge on identifying the RCs. RCs are the cornerstone of the standard reaction rate theories. Reaction dynamics is the dynamics of RCs. Kramer's theory is based on the assumption that the dynamics of the RC is governed by a Langevin equation, 4, which is extended to generalized Langevin equation in Groth-Hines theory, 5. Similarly, transition state theory relies on simplifying assumptions on the essential features of the dynamics of the RC, 6 to 10. Applicability of all these theories requires full knowledge of the RCs. Understanding reaction mechanism amounts to answering 4 questions. 1. What are the RCs? 2. How does a molecule move along its RCs? 3. What are the physical interactions that determine the RCs? 4. How do these interactions control the RCs? Aside from their fundamental theoretical value, RCs are central to 2 major topics in computational biophysics. 1. Dimensionality reduction on large-scale simulation data, and 2. Enhanced sampling. Advances in computational power have enabled simulation of protein dynamics in the range of hundreds of microseconds to milliseconds, either as an accumulation of a large number of short dynamic trajectories as routinely done in Markov state modeling and weighted ensemble simulations, 11, 12, or in a single long trajectory using a specialized machine such as Anton, 13. The purpose of these simulations is to build mechanistic and kinetic models of functional processes, which is extremely challenging because of the curse of dimensionality. The key to overcome this difficulty is to identify a small number of collective variables, CVs, that can capture the essence of protein dynamics. By definition, RCs provide the optimal dimensionality reduction. The purpose of enhanced sampling is to bridge the gap between timescales easily accessible to molecular dynamics, MD, simulations, for example, 100 ns, and timescales of functional processes, for example, milliseconds to seconds. Most algorithms, such as umbrella sampling, 14, metadynamics, and adaptive biasing force, 15 to 18, rely on applying bias potentials on one or a few selected by intuitions to increase sampling of the relevant configuration space. The efficacy of an algorithm critically depends on the quality of the CVs. Only CVs that significantly overlap with the RCs are effective. Otherwise, the infamous hidden barrier problem will inevitably appear and prevent effective sampling. 1920. The hidden barrier is the actual activation barrier that lies on the true RCs, which will be missed by the bias potentials if CVs do not have significant overlap with the RCs. Developing systematic methods for identifying CVs that can improve sampling efficiency has attracted intensive research activities in recent years. 21 to 23. The rigorous concept of RC. Given the importance of RCs, a rigorous definition is essential. A central concept is committer. The probability that a dynamic trajectory initiated from a system configuration, with initial momenta drawn from Boltzmann distribution, to reach the product state before the reactant state. 3, 24 to 26. Committer rigorously parameterizes the progress of a reaction, for example, the transition state has. Consequently, RCs are the set of coordinates that can fully determine the committer value of any system configuration. Other coordinates in the system are irrelevant. RCs that satisfy this definition provide the optimal reduced description of a reaction that captures the essence of the reaction dynamics. In contrast, ad hoc RCs that do not satisfy this definition bear little relevance to reaction dynamics and mechanism. 27, 28. Challenges in identifying RCs. Du et al. First clarified the importance of RCs to protein folding in their pioneering paper in 1998, 24. Chandler and co-workers systematically established the importance of identifying RCs in activated processes in general in a series of seminal works in early 2000s. 3. Since then, identifying exact RCs that can determine committer in complex molecules has been a central topic in theoretical chemical physics and biophysics. It turned out to be a formidable challenge. Despite intensive efforts over the past 2 decades, there have been only a few successful cases where exact RCs were identified, 29 to 32. In each case, 2 or 3 RCs were successfully identified. The most important lesson from these examples is that RCs are often counterintuitive, 29, 30. This counterintuitiveness demonstrates why it is so challenging to identify the few RCs out of practically infinitely many potential candidates in a complex molecule. For example, over 6,000 candidates were tested in Ref. 30 for the isomerization of an alanine dipeptide in solution to uncover the 3 RCs. As a result, systematic methods that go beyond intuition-based trial and error experimentation are required, because it is not feasible to enumerate the vast number of potential candidates. In 2005, Ma and Dinner, 30, used neural network to successfully identify the RCs for the isomerization of an alanine dipeptide in solution that eluded human intuitions, 33. This early success was followed by many machine learning methods along similar lines, a research direction that is currently attracting intensive attention but also facing significant challenges. 34-39. More importantly, machine learning methods cannot answer the central question concerning RCs. Why do RCs exist and how do they control functional dynamics? Answering this question requires the correct mechanical model of functional dynamics, which machine learning cannot provide because it can only optimize parameters based on a pre-formulated model but cannot devise a model de novo. For this purpose, an approach based on the principles of fundamental physics is required. In Ref. 40, we developed a fundamental mechanical operator, the generalized work functional, GWF. Applying the GWF to the transition of an alanine dipeptide in vacuum, a prototype of biomolecular isomerization dynamics and standard testing case for RC identification, we successfully identified the exact one-dimensional RC as the leading left singular vector of the GWF. In the current manuscript, we applied the GWF method to an important functional process, the flap opening of HIV-1 protease, HIV-PR. Unlike the case of alanine dipeptide, where the transition process is one-dimensional with only one RC, we found that the flap opening process is intrinsically multidimensional, with 6 RCs corresponding to the 6 leading singular vectors of the GWF. Each RC is a linear combination of 240 backbone dihedrals and precisely defines the global cooperativity of this process. Applying bias potentials on these RCs leads to flap opening within picoseconds, in contrast to the tens of nanoseconds required for straightforward MD simulations. An important feature of the GWF method is that it is firmly based on physical principles rather than relying on intuitions. Methods of this nature have a long history, dating back to the variational transition state theory, 41. They include the theory of normally hyperbolic invariant manifold, 43, the method of minimizing variations at the stochastic separatrix, 44, and methods that maximize reactive flux, 45-47, or minimize reaction time, 48. The Thurch, Verse, and Pers, Thasher, 9. This GWF method uses a rigorous operator of classical mechanics to construct global and collective RCs of a functional process in a large protein directly from elementary coordinates that describe local movements of the protein. We show that these RCs can determine committer with high accuracy. Results. We first summarize the major theoretical tools and then present our results on the HIV-PR energy flow theory. A major theoretical tool for analyzing reaction dynamics is the energy flow theory, 49. It was motivated by a fundamental physical consideration. Motions of the critical coordinates in a dynamic process invoke high energy cost. In the Lagrangian Hamiltonian formulation of classical mechanics, energy is the cost function of motion. If the motion of a coordinate invokes high energy cost, then it has a slow timescale and controls the rate of a reaction process. It is likely a RC. To identify these critical coordinates, we need a rigorous definition of the energy cost of the motion of a coordinate. This is challenging because the energy function of a system is dominated by complex coupling terms between different coordinates. There is no well-defined energy per coordinate, making it difficult to define energy cost per coordinate. This conceptual and practical difficulty can be resolved by realizing that the total differential of a general multivariable function is, thus each partial differential is the exact contribution to the total change in, due to the change in, the coupling terms in, however complicated they may be, are precisely partitioned among all the variables through partial derivatives. Consequently, rigorously defines the contribution to the accumulated change in, due to the change of, from, to. Applying this idea to the potential energy of the system, we define the potential energy flow, PEF, of a coordinate. as the mechanical work done on 31. 1. Where is the potential energy of the system and is the position vector of the system in the configuration space, according to equation 1, is the change in caused by the motion of alone along a dynamic trajectory in the time interval. It is a projection of the change in the total potential energy onto and a measure of the cost of the motion of. Kinetic energy flows can be defined in a similar manner. 49. To gain mechanistic insights, we need to look at how a mechanical quantity, for example, PEF, changes as a reaction progresses. We first project onto a variable that parameterizes the progress of a reaction, which we call the projector, then average over the ensemble of reactive trajectories. 2. Here is the probability of finding the system in an infinitesimal volume around a phase space point in the reactive trajectory ensemble is the Dirac delta function, is the change in over a differential interval, semicolon, is the change in over a finite interval, semicolon, can be 31. 49. The ensemble of reactive trajectories consists of trajectories that cover the transition period of a reaction process, but exclude the waiting period. 3. In Ref. 40, we showed that an order parameter that can distinguish the reactant from the product state can serve as the projector for identifying the RC's GWF. In Ref. 40, we developed a fundamental mechanical operator, the GWF, that can be used to identify the RCs based on the principles of class mechanics alone. The differential definition of the GWF is 3. Here is the GWF is the vector of infinitesimal displacements in coordinate system and is the corresponding force vector. We termed the GWF because its element is a generalization of the concept of mechanical work. It is an asymmetrical tensor, and each diagonal element is the mechanical work done on coordinate. It is an extension of the mechanical work because the total work done, i.e., the change in system potential energy, on the system, can be obtained from through tensor contraction. 4. In contrast, cannot be computed from. Importantly, is the kernel for the transformation of between different coordinate systems. As an example, we consider two coordinate systems and related by and where and its inverse are the coordinate transformation matrices. If we know the work in where, then we can compute the work in through coordinate transformation and obtain where means the vector formed by all the diagonal elements of matrix. These results suggest that contains critical information of the mechanical effects of forces in a system that is not included in the mechanical work. In Ref. 40, we found that the leading left singular vectors of the GWF, which we termed the singular coordinates, are the exact RCs. Singular vector decomposition reveals the intrinsic structure of the collection of all the column vectors of, i.e., the left singular vectors, where is the eighth column of, forms an orthonormal basis of the vector space of. Similarly, the column vectors of form an orthonormal basis of the vector space of. Because a singular value indicates the impact of the force along the direction of on the dynamics of the system, a large value of means has high impact because a functional process has a dominant direction. For example, from the inactive to the active conformation, the direction of the RC, the impact of a force on the system mainly reflects its impact on the RC. Therefore, a force with high impact on the system has a high impact on the RC, making the direction of this force the direction of the RC. The importance of each singular coordinate is ranked by its corresponding singular value, which is also proportional to its PEF. This is the physical reason that the leading singular coordinates of the GWF are the RCs and their relative importance is ranked by their singular values. To compute the RCs, we use the left singular vectors of the ensemble averaged GWF. Where is the projector? In general, a functional process is parameterized by over an interval. This interval is divided into many small intervals of size, and we compute from For each interval, the concatenation of all provides a piecewise linear approximation of as a function of 40. In this way, we obtain a piecewise linear approximation of the true RCs. Although the RCs can be complicated nonlinear curves in the configuration space, an RC in an interval can always be approximated by a linear function if is sufficiently small. Therefore, piecewise linear approximation with sufficiently small can provide sufficient accuracy no matter how nonlinear the RCs are. In practice, if is a well-behaved function of then obtained from the GWF over an extended interval. 5 is a good approximation to the true RC. Because integration is related to averaging by a simple scaling factor, has much lower noise than this can lower the computational cost tremendously. Therefore, in practice the RCs for a real system are computed from instead of flap opening of HIV-PR. In the current paper, we aim to identify the exact RCs for the flap opening of HIV-PR. Due to its critical role in processing viral polypeptide precursors, HIV-PR has been a major drug target for several decades. 50. It is a symmetric hemidimer. Each monomer has 99 residues. 2 glycine-rich flaps, figure 1, serve as the gating mechanism that controls a substrate's access to the active site. Based on previous experimental and computational studies, the flaps can adopt 3 conformations: closed, semi-open, and open, 51 to 53. While semi-open is dominant for substrate-free HIV-PR and closed is dominant for substrate-bound HIV-PR, transition from semi-open to open is necessary for substrate binding. Understanding the molecular mechanism of this process is critical for understanding the mechanism and pathway of substrate binding, which is critical for rational design of effective inhibitors of HIV-PR. 54. A critical step in understanding the mechanism of flap opening is to identify the exact RCs that can determine the committer value. Figure 1. Representative structures of the semi-open and open state of HIV-PR. The semi-open structure is from Crystal Structure, Protein Databank, 1HHP, and the open structure is from TPS trajectories. Different structural segments are marked by different colors. The 2 spheres represent the center of mass of the atoms of residues met 46 to Lys 55 in the 2 monomers, which are used to define the flap distance. Following the simulation procedure by Hornak et al. 52, we used straightforward MD to simulate the system in implicit solvent for 30 Ns. We observed a transition from semi-open, the starting crystal structure, to open at 21 Ns. Using the trajectory segment containing this flap opening event as the initial seed trajectory, we used transition path sampling, TPS, to proliferate reactive trajectories for the flap opening process. 3. In total, we obtained 2,000 reactive trajectories, which are used in the analyses below PEFs. The PEFs of internal coordinates, ICs, of HIV-PR have distinct features compared to the PEFs of ICs of smaller systems such as alanine dipeptide. In the alanine dipeptide case, the PEFs separate into 2 groups, figure 2 in Ref. 31. One group consists of the 2 major RCs, dihedrals, and the other group contains all the other 58 coordinates. The PEFs of the former have much higher magnitudes than the PEFs of the latter, distinguishing the former as the RCs. Moreover, we observed, and along almost all the reactive trajectories, showing that needs to cross an energetic barrier while receives energy from the non-RCs, which is then used to assist in barrier crossing. 31, 49. In HIV-PR, however, the PEFs of ICs show a continuous distribution. There is no clear separation in the magnitudes of the PEFs that distinguishes the RCs from the non-RCs, figure 2a. Moreover, for each coordinate, we observed, along some trajectories and along the other trajectories, these 2 groups of trajectories are of similar numbers. This situation indicates that, in HIV-PR, the RCs involve many ICs and are global and cooperative. To decipher the information embedded in the PEFs, for each, we divide all the reactive trajectories into 2 ensembles. Consists of all the trajectories with, and consists of all the trajectories with. For each and contain roughly the same number of trajectories, i.e., 1,000 trajectories each, for two coordinates, and, in general, and to compute the average PEFs, we use the distance between the two flaps as the projector, which is defined as the distance between the center of mass of residues met 46 to Lys 55 in each monomer, figure 1. We choose this definition so that is insensitive to fluctuations in the flap tips, which are highly flexible. Figure 2. PEFs of internal and singular coordinates and the GWF, A, PEFs of all the backbone dihedrals averaged over their respective, and each PEF curve is colored based on the structural segment that belongs to B, box and whisker plot of the PEFs of backbone dihedrals in each structural segment at NM and averaged over C, PEFs of all the singular coordinates averaged over corresponding, and thick colored lines are PEFs of 2, D, GWF of all the backbone dihedrals calculated for. Figure 2a shows the PEFs of all the backbone dihedrals averaged over there, and, respectively, for each dihedral. Each PEF curve is colored according to the structural segment that belongs to, for example, if belongs to the flap, then both, and are colored red. Dihedrals with high PEFs are mainly in the flap, elbow, and cantilever segments, figure 2b. Aside from the magnitude, the PEFs of all backbone dihedrals have similar profile. They rise quickly in the interval and start to plateau after NM, suggesting that the system needs to climb up a steep energetic barrier first and the dynamics becomes mostly diffusion-like afterward. GWF and singular coordinates, figure 2d shows, calculated for all the backbone dihedrals. Forces on dihedrals and structural segments with high PEFs, such as flap, elbow, and cantilever, have high impacts on the motions of most coordinates in the system. In contrast, forces on dihedrals and segments with low PEFs, for example, active site and interface, have little impact. Figure 2c shows the PEFs of all the singular coordinates from. In contrast to the PEFs of ICs, the PEFs of singular coordinates separate into two distinct groups. A small number of singular coordinates, 2, have PEFs higher than the highest PEFs of the ICs, and their PEFs are well separated from each other. In contrast, PEFs of the other singular coordinates are significantly lower and show a dense and continuous distribution. This result shows that forces along 2 have high impact on the dynamics of the system, whereas the other singular coordinates do not, making 2 the natural candidates for the RCs. An interesting case for comparison is the PEFs of singular coordinates for isomerization of the alanine dipeptide, where only has high PEF, while all the other singular coordinates have vanishing PEFs. Therefore, while the isomerization of alanine dipeptide is a one-dimensional process with only one RC, the flap opening of HIV-PR is intrinsically multidimensional with 6 RCs. Each singular coordinate is a linear combination of 572 backbone dihedrals, where are elements of the left singular matrix from singular value decomposition, SVD, of. Each provides a natural description of local movements of the protein backbone and precisely defines the cooperative relation between different. Therefore, RCs obtained from the GWF not only are global and collective but also define the precise meaning of cooperativity. This is in stark contrast to CVs constructed by intuitions, for example, which are collective, but it is unclear how the complicated collective motions are realized through aggregation of local motions that are the elementary steps of protein motion. Therefore, these CVs are less useful in understanding mechanisms even if they happen to be good RCs. Figure 3 shows the coefficients for 2. Dihedrals with large coefficients are located in the flap, elbow, and cantilever region, which are also regions with high PEFs, indicating that the strong cooperativity among these coordinates is due to the strong interactions and mechanical couplings among them. Figure 3. Coefficients of singular coordinates. 2. Dihedral indices for different structural segments are marked by different colors in the top panel. Physical picture of an intrinsically multidimensional process. Based on the results above, The activation barrier of the flap opening of HIV-PR has an extended topology that resembles the rim of a crater, instead of the saddle point commonly assumed in multidimensional theories of reaction dynamics. 7.55. Figure 4 shows the schematic free energy landscapes for these two scenarios. For the case of a saddle-like barrier, Figure 4a, all the reactive trajectories will be aligned with the sole unstable direction at the barrier top. Consequently, a one-dimensional RC can be constructed from the multidimensional RCs so that the rate predicted by the one-dimensional Kramer's theory will be identical to Langer's multidimensional result, as shown in the seminal work by Bereshkovsky and Szabo. 55.56. In this case, it is possible to replace the multidimensional RCs by a one-dimensional RC that corresponds to the single preferred direction at the barrier top. Figure 4. Schematic free energy landscapes for intrinsically one-dimensional, panel A, and two-dimensional, panel B, processes. In A, the yellow curve denotes a typical reactive trajectory. The two dashed arrows indicate the directions of the multidimensional RCs, and the solid black arrow indicates the direction of the one-dimensional RC that can produce the same reaction rate as the multidimensional RCs. In B, the red curve denotes a typical reactive trajectory. In contrast, for the case of a rim-like barrier, Figure 4b, the enthalpy-stabilized reactant basin, for example, there are many strong interactions between the two monomers of HIV-PR in the closed structure, is separated from the broad entropy-stabilized product basin. For example, there are much less interactions between monomers in the open structure, so they can sample a much larger region in the configuration space by a broad enthalpic barrier. For example, breaking the monomer-monomer interactions requires significant energy. In this case, the reactive trajectories no longer have a single preferred direction even at the barrier top region. A trajectory that strictly follows the direction of an RC will indeed be a reactive trajectory, but a typical reactive trajectory rarely follows a single RC. Instead, each reactive trajectory traces out a meandering curve that is a nonlinear and stochastic function of all the RCs. Figure 4b. Collectively, the set of RCs form an orthonormal coordinate system that provides the optimal description of the transition region. In this case, the multidimensional RCs cannot be replaced by a one-dimensional RC. Committer test on the singular coordinates. To determine if 2 are indeed the correct RCs, we need to test if they can determine the committer value for any system configuration. The standard committer test involves generating an ensemble of structures that all share the same values of the RCs that correspond to the transition state, i.e., but differ in the other coordinates. 3.24. If the distribution of committer values of this ensemble of structures is centered around, then it indicates that the RCs can determine the committer value. Conducting committer test requires knowledge of the relationship between the RCs and committer so that we can find the RC values corresponding to. This is not an issue for processes with only one RC, in which reactive trajectories from TPS simulations all move along the direction of the RC. We can identify the RC-committer relationship by inspecting the committer values of configurations along a TPS trajectory. This strategy no longer applies when there are multiple RCs because each TPS trajectory, instead of following one RC, will trace out a curve in the configuration space that is a nonlinear and stochastic function of all the RCs. It is exceedingly difficult to decipher the RC-committer relationship from such curves. Moreover, committer calculations are very costly for large systems like HIV-PR, so we cannot compute committer values for many configurations. This lack of data will make it even more formidable to extract the RC-committer relation from TPS trajectories. Fortunately, another critical feature of the RCs provides a solution to this difficulty. RCs are the most efficient directions for the system to move from the reactant to the product state, which is the reason that RCs are considered the most effective CVs for applying bias potentials in enhanced sampling algorithms. 22. This notion is confirmed by the meaning of RCs as the forces with the highest impact on the reactive dynamics based on the GWF theory, which suggests that, if we apply a bias potential in the direction of an RC, the system will move toward the product state with the highest efficiency. The PEFs of RCs provide a ranking of the efficacies of different RCs. Forces acting on an RC with higher PEF are more effective in moving the system toward the product state. Consequently, if 2 are the RCs, we can apply bias potentials along each. The resulting trajectories should move preferentially along, and the system should move toward the open state with high efficiency. From such a trajectory, we can obtain the RC-committer relationship for. Figure 5a shows the time dependence of along trajectories obtained from applying bias potentials on. Details of the bias potentials are in the methods section. In all cases, the system will undergo semi-open to open transition within 20 Ps. The efficiency for opening up the flaps decreases from 2, consistent with their rankings based on, and is a comparison. Figure 5b shows the time dependence of along reactive trajectories from TPS simulations. The starting point of each TPS trajectory is chosen as the last time that equals to 1.3 nanometers along that trajectory. Thus figure 5b shows the fastest time that reaches a specific value along each TPS trajectory. Since a TPS trajectory only contains the transition period but excludes the waiting time, 3 results in figure 5b represent the highest efficiency that the natural proceeding of flap opening can reach under the current force field. Figure 5. Comparison of the efficiency of flap opening along biased and TPS trajectories. A. Time dependence of along trajectories generated from applying bias forces on. 2. Solid lines are results of averaging over 20 trajectories for each. The red shade marks the range covered by biased trajectories along. The lower bound is the lowest value, and the upper bound is the highest value among all the 20 trajectories at each time point. The gray dashed horizontal line marks NM. B. Time dependence of along TPS trajectories. The solid line is the average over all 2000 TPS trajectories. Dark blue shade marks the range covered by one standard deviation. Light blue shade marks the range covered by TPS trajectories, defined the same way as in the left panel. The vertical dashed line marks Ps. The blue dashed horizontal line marks NM. The red dashed horizontal line marks NM. The bias trajectories are significantly more effective than TPS trajectories in opening the flaps. On average, the least effective RC, i.e., can open the flaps to NM by the end of 20 Ps, while the most effective trajectories along can reach NM. In contrast, TPS trajectories on average need 120 Ps to reach NM. For the most effective RC, i.e., on average the flaps open to NM by 20 Ps, which is beyond the highest efficiency of TPS trajectories. The most effective trajectories along can reach NM, far above the highest efficiency by any TPS trajectory. These results show that applying bias forces on 2 can open the flaps with an efficiency that is above the upper limit observed in TPS trajectories, suggesting that they represent the upper limit for flap opening. This strongly suggests that they are indeed the correct RCs. Moreover, provides accurate ranking of the efficacy of confirming their physical meanings defined by the GWF. In the current implicit solvent model, flap opening, i.e., reaching NM, takes tens of nanoseconds in straightforward MD simulations, a result consistent with observations by Hornock et al. 52. By excluding the waiting time, on average this time scale is reduced to 100 Ps in TPS trajectories. In contrast, biased trajectories from applying forces on the RCs only need 4 or 10 Ps. 2. To open the flaps, amounting to fold acceleration over straightforward MD simulations. This is achieved by more efficient energy flows into the RCs and reduction in diffusive motions and stochasticity because we directly do mechanical work on the RCs via the bias potential. Figure 6 shows the open structures from biased trajectories along 2. The conformational changes are mainly in the flaps, elbows, and to a lesser extent the cantilevers and fulcrums, while the monomer interface remains intact, similar to structures from TPS trajectories. Different singular coordinates open the flaps along different directions, which reflects their orthogonality and is consistent with the diverse ways of flap opening we observed in the TPS trajectories. This further confirms the multiplicity and multidimensional nature of the flap opening process. Figure 6. Representative open structures from biased trajectories along. 2. The reference semi-open structure. NM is the starting structure of the biased trajectories and is colored white. In the open structures, atoms of each backbone dihedral are colored according to its coefficient N. Green, red, blue. Darker red or blue color indicates higher magnitude of. Figure 7a shows the committer values of configurations along trajectories obtained from applying bias forces on 2, respectively, in each case, is a sigmoid function of, which is typical for the committer-RC relationship, 37. To do the committer test, we generated additional trajectories by applying bias forces on 2. From each trajectory, we harvested configurations that should have, based on the results in Figure 7a and computed their committer values. Details in the methods section. The resulting distribution is shown in Figure 7b, which shows a Gaussian-like profile peaked at, confirming that 2 are indeed the correct RCs. This result shows that the GWF provides a general, rigorous, and physics-based method for identifying the RCs in complex molecules. Figure 7. Committer test for 2. A. The value of committer as function of. Along biased trajectories, the error bar for each committer value is calculated using the formula from Ref. 57. In each subpanel, the range of, marked by the two vertical dashed lines, corresponds to. Additional bias trajectories are generated to harvest system configurations with, in these ranges, B. Committer distribution over the ensemble of system of configurations that are predicted to have. Based on their. It is worthwhile to make a comparison with principal component analysis, PCA, 58, 59, which is widely adopted for analyzing structural dynamics of proteins and identifying CVs. The PCs are eigenvectors of the covariance matrix of system coordinates with large eigenvalues. They are the directions that show large variations during the process under study. In previous applications of PCA to the conformational dynamics of the alanine dipeptide, however, it was found that PCs cannot determine committer 30, 33, 60. Thus they are not the RCs. The fundamental difference between GWF and PCA is the matrices they use. The covariance matrix is based purely on the movements of coordinates, which are observables of the equations of motion, EOM. In contrast, the GWF tensor is directly related to the potential energy function, which is the generating function and thus the determinant of the EOMs, because the EOMs are obtained from taking derivatives of the potential energy function. The GWF is related to the potential energy in two ways. 1. The change in system potential energy is related to the GWF by tensor contraction. And 2. Is the kernel for the transformation of the mechanical work, i.e., between different coordinate systems. As a result, while PCA is based on the observables of, the GWF is based on the determinant of the EOMs, which is the reason that the GWF can identify the exact RCs discussions. In the current paper, we applied the recently developed GWF method to the flap opening process of HIV-PR. We found that the 6 leading left singular vectors of the GWF are the exact RCs that can determine the value of committer for any system configuration. This is the first time that the exact RCs are identified for a major conformational change of a large functional protein. Comparing to other existing methods for identifying the RCs, 30, 34, 36, 39, the GWF method has the following advantages. 1. The GWF method directly calculates RCs from a rigorous operator of classical mechanics, rather than uses machine learning to pick out coordinates that can meet empirically defined mathematical criterion, for example, strong correlation with committer values across the training set, from a pool of candidates prepared by intuitions. 2. Section 2. The GWF method does not require any input from human intuition or committer information and handles different systems, for example, HIV-PR versus alanine dipeptide, in a uniform manner. These facts show that the GWF provides a physics-based method for identifying the RCs in complex molecules that is rigorous and general. Applying bias forces along the directions of the RCs accelerates flap opening by 103 to 104 folds compared to straightforward MD simulations, and 10 to 25 folds compared to TPS trajectories, which represent the limiting efficiency of the natural proceeding of this process. This confirms the long-held assumption that applying bias potentials along the exact RCs provides the ultimate efficiency for enhanced sampling. Finally, RCs identified by the GWF method are linear combinations of ICs, each one providing the natural description for local protein motions. Therefore, RCs identified by GWF provide the precise physical meaning of collectivity and cooperativity of protein dynamics. Together, these results suggest that the GWF is likely the fundamental physical operator that controls the systematic functional dynamics of molecular systems. Finally, we want to discuss our perspective on potential applications of the GWF method to other important problems, such as protein folding, entropic barrier, and reaction rate. Protein folding is a central topic in biophysics. Identifying its RCs is critical for understanding its mechanism in detail. According to the energy landscape theory, folding consists of a series of conformational changes from the unfolded toward the more folded structures via multiple parallel pathways. 61, 62. Conformational changes during folding have an additional complexity compared to those of a functional process because they involve transitions from multiple reactant basins, i.e., multiple ensembles of highly diverse unfolded structures, instead of one reactant basin, to the product basin, i.e., the folded structure. We expect that the GWF method can identify the RCs for folding based on the following reasoning. Successful applications of the GWF method rely on two factors. 1. The process of interest must have a direction of progress, and 2. We can find an order parameter that can roughly capture this direction. For example, it can clearly distinguish the reactant basin from the product basin. This order parameter is used as the projector in the ensemble average in equation 2, which eliminates noise and reveals the underlying systematic dynamics along the RCs. The number of reactant basins and the diversity of the structures corresponding to different basins are not important, as long as their transitions toward the product basin share a common direction that can be adequately described by an order parameter. Folding has a clear direction, toward the more folded structure. This direction can be captured by the number of native contacts or the fraction of residues in the native configuration, based on the analyses of long folding trajectories by Linderflarsen et al. And by Best and Hummer, who showed that these parameters can adequately characterize the metastable basins, i.e., folding intermediates, of folding. 1363. The transitions from different unfolded structures likely have different RCs, which will likely show up as separate clusters of singular coordinates from SVD of the GWF. Each cluster represents the RCs for the transition from one unfolded basin to the folded basin. Each unfolded to folded transition will likely have multidimensional RCs similar to the flap opening process of the current study. Entropic barrier is an important mechanistic model for protein conformational changes and folding. 64. In this case, the RCs are the directions pointing from the reactant basin to the location of the barrier. Because the barrier is entropic, there is no systematic forces that push the system toward or away from the barrier. Instead, all the forces in the system act in random directions. At first sight, it may appear that this defies the very strength of the GWF, which, in the case of enthalpic barriers, identifies RCs by identifying forces that have systematic effects on the system. However, for dynamic trajectories that successfully reach the entropic barrier, the overall effects of all the forces acting on the system must be along the directions of the RCs. The GWF should be able to capture these directions because the forces that point to the other directions are completely random and their effects will be averaged out by the ensemble average. The only forces left with non-vanishing effects are those along the directions of the RCs. Thus, we expect that SVD of the GWF should still yield the correct RCs of an entropic barrier. Aside from understanding mechanisms, a major usage of RCs is to compute reaction rates. Because the RCs identified by the GWF method can determine committer with high accuracy, they satisfy the criteria for the RCs in standard reaction rate theories. Since the dynamics of conformational changes are likely diffusive, a multidimensional version of Kramer's or Grotein's theory will be the proper choice. 4, 5, 55. However, Langer theory, the multi-dimensional extension of Kramer's theory, assumes that the top of the activation barrier resembles a saddle point, while our results on flap opening suggest that the barrier top more likely resemble the rim of a crater. Therefore, we expect that an extension of the Langer theory to properly handle rim-like barriers will be required for accurately computing reaction rates. All simulations are performed using the Amber 99C force field with the Onufrev-Bashford case, OBC, implicit solvent model, 65, and the Groningen machine for chemical simulations, GROMACS, 4.5.4 simulation suite, 66, 67. To generate the initial reactive trajectory for the flap opening process, we conducted a 30 ns-MD simulation following the procedure by Hornock et al. 52. The initial structure of HIV-PR is from the crystal structure of the semi-open conformation protein databank, 1-HHP, 68, with 2 mutations, EF and C95A. Residue ASP25 is modeled in the protonated state. The simulation was carried out in the canonical ensemble, and the temperature is maintained at 300 K using Langevin dynamics with a friction constant of . All the bonds involving hydrogen atoms are constrained using the Shake algorithm, 69. Time step used in simulations is 1 fs. Within the 30 ns simulated time, we observed 1 instance of flap opening. The trajectory segment that contains this event is used as the seed trajectory for subsequent TPS simulations. In TPS simulations, we define as the semi-open state and as the open state. Each TPS trajectory has a length of 120 ps, and the acceptance ratio of shooting moves is 0.18. 3. Bias trajectories along 2. 2. Test the quality of singular coordinates. 2. As RCs, we applied bias forces on them to see if the flaps will open at an accelerated timescale. Because of the periodicity of angles, we replace by 70, where is an element of the left singular matrix of the GWF. Is the number of coordinates included in the definition of is a backbone dihedral. Is the target value of is the value of in the starting structure, and is the constant that we use to decide how many units of we want to move the system. The use of cosine function is to remove the discontinuity caused by the periodicity of angular coordinates. Because the GWF inevitably contains noise, we do not include all 572 backbone dihedrals in . Instead, we only include if to remove backbone dihedrals that are included in due to the noise in the GWF. In current calculations, we used resulting in . To apply the bias force gradually, we divide the interval between the minimum value and the target value of into 100 bins, with the nth bin spanning the interval. At any instant t, when the system configuration is at it feels an bias potential of the form . In this way, the system always feels a gentle force pulling it toward the target value, with the center of the bias potential shifting adaptively as the system moves from one bin to another. For the committer test, we first decide the range of that correspond to for each . On each committer relationship curve in Figure 7a, the range between the 2 vertical dashed lines corresponds to . We then generate more biased trajectories along each and harvest configurations with. In these ranges, the committer for each configuration is then calculated, and the resulting distribution is plotted in Figure 7b. The error bars for committer values in Figure 7a are calculated using the formula where is the number of trajectories initiated from a system configuration in the calculation of its committer value. Is the committer value calculated using trajectories, and is corresponding error bar, 57. Data, materials, and software availability. All study data are included in the articled acknowledgements. We thank NIH, R01 GM08 6536. R21 AI 160 2197, and NSF Award, CHE 1,665,104, for financial support. We thank Stuart Rice and Attila Szabo for their critical reading of an early version of the manuscript and insightful comments. We also thank the anonymous reviewers for their excellent suggestions that helped to improve the manuscript. This research used resources of the Argonne Leadership Computing Facility, which is a DOE Office of Science user facility supported under Contract DAC 0206-11357. Author contributions A.M. Designed research, SW, HL, and AM performed research, SW, HL, and AM analyzed data, and AM wrote the paper. Competing interests: the authors declare no competing interest. Exact reaction coordinates for flap opening in HIV-1 protease. Shanshan Wu, Huiyu Li, https://